citric acid cycle is a series of chemical reactions that takes place in the mitochondrial matrix. This cycle uses acetyl-CoA derived from sugar and fat breakdown to form ATP, NADH, FADH2, and carbon dioxide. The NADH and FADH2 can be used to form additional ATP through the electron transport chain. The citric acid cycle goes by many names, including the tricarboxylic acid, or TCA cycle, and the Krebs cycle. Citric acid refers to the citrate that is produced in the first step of the pathway. The tricarboxylic acid title gets its name from the 3 carbon dioxides that are produced for each fully oxidized pyruvate. Krebs refers to Hans Adolf Krebs, who identified the full cycle in 1937. He was eventually awarded the Nobel Prize for Physiology or Medicine in 1953 for his discovery. In the first step of the cycle, an enzyme called citrate synthase joins the 2-carbon acetyl group from acetyl-CoA with the 4-carbon oxaloacetate to form a 6-carbon citrate. In step 2, an enzyme called aconitase converts citrate into isocitrate. Next, an isocitrate dehydrogenase enzyme oxidizes isocitrate, a 6-carbon molecule, into a 5-carbon alpha-ketoglutarate. The carbon that was lost is released as carbon dioxide, and 1 NADH is also formed. The carbon dioxide that is released was originally part of oxaloacetate and not acetyl-CoA. In the fourth step, an enzyme called alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate into a 4-carbon succinyl-CoA. Similar to step 3, this reaction produces 1 carbon dioxide and 1 NADH. In step 5, a succinyl-CoA synthetase enzyme converts succinyl-CoA into succinate. This produces GTP, which is converted to ATP. In step 6, an enzyme called succinate dehydrogenase converts succinate into fumarate. This step makes 1 FADH2. In step 7, a fumarate hydratase enzyme then converts fumarate into malate. In the final step of the citric acid cycle, a malate dehydrogenase enzyme converts malate back to oxaloacetate. Like all steps involving a dehydrogenase, a coenzyme is produced. Here, it is NADH. The oxaloacetate that was regenerated through the citric acid cycle is now ready to join with another acetyl group and begin the cycle a second time. For every 1 glucose that is broken down through glycolysis, 2 pyruvates will be produced. These 2 pyruvates will produce 2 acetyl-CoAs. So for every 1 glucose, 2 acetyl-CoAs will be made and 2 turns of the citric acid cycle will occur. This means each product of the cycle must be doubled. A total of 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs are made through the citric acid cycle. NADH and FADH2 are electron carriers that can produce more ATP later in aerobic respiration. In addition to sugars like glucose, proteins and fats can also provide carbon substrates to fuel the citric acid cycle. Proteins can be broken down into individual amino acids such as alanine, aspartate, and glutamate, and converted into intermediates in the cycle. Fatty acids can be broken down into acetyl-CoA, which then begins the citric acid cycle. This metabolism of sugars, proteins, and fats through the citric acid cycle provides the vital energy necessary to maintain many cellular processes.